Thank you so much for that kind and flattering introduction. I'm really sorry not to be able to present this lecture in person. I suppose this is poetic justice for having myself been involved in studying the COVID-19 spike protein, shown here with this glycan shield on the right side of the front cover. Adding my image and name to the slides is a surreal experience, and I truly feel like an imposter in the midst of this constellation of superstars in biochemistry and molecular biology. In this short talk, I'd like to trace the origins of my interest in biological chemistry, focusing on work that was largely published in JBC. I had the good fortune of having acquaintance with Herbert Tabor over the years. And one thing that particularly helped me was the conversion to online publishing of the Journal of Clinical Investigation as editor. But actually, it was the first journal to come free online in 1996. Actually, I beat him to it. But his advice was extremely helpful in giving this free online access. As part of my interest in peer review, I also found that I felt there was a need for uniform code of ethics for editors of peer-reviewed biomedical journals. Again, Herbert was supportive of me and gave advice, but in the end, I could not get the editors of major journals to participate. I think they like their power, basically. Going back in time, I began my medical education at CMC Vellore, a remarkable institution in southern India, just started by Ida Sophia Scudder, the first female graduate of Cornell Medical College. She started the medical school and then led men in 1949. She wanted a medical school for women primarily. Also at CMC, I met my spouse and long-term partner, Ms. Lonan, without whom I wouldn't be here. And in fact, I think this prize goes as much to her as to me. The biochemistry department at CMC Vellore offered a prize for the best medical student essay on a topic that is biomedical importance. It happened that the 1970 Nobel Prize in Medicine and Physiology was awarded to Earl Wilbur Sutherland, an American physician who discovers the hormonal action, particularly isolation of cyclic AMP. Yes, I read more about this, not part of a regular curriculum, of course. As you can see here, I found that the mechanism of cholera fluid production in the intestine is related to cyclic AMP, and so there's definitely a biomedical relevance to this. In looking through these papers, I found amazingly batches of papers together that related, and I wish someday I could do this. And it was really remarkable to see this. I then realized that why the North American medical students medical schools seem to be doing a lot more research. Flexner report in 1910 required all practice of medicine to ground themselves in fundamental science upon which medicine rests. This is the origin of the trigger of scientific application to medicine. It happened that the U.S. Immigration Naturalization Act of 1965 abolished quota systems based on national origins. New policy was based on reuniting immigrant families and attracting skilled labor to the United States. At that time, foreign medical graduates like myself were invited to staff hospitals, communities lacking physicians in exchange for legal immigrant status. I was not interested in going to private practice, but I took advantage of this program to gain access to the U.S. as a legal immigrant, arriving $6 in a suitcase and working my way up the system. I was originally interested in studying blood cells and bone marrow transplantation, as Jamie mentioned. But as I look further, I realized that every cell in the body is covered with all the blood cells are covered with a big, thick array of sugars called glycocalyx, and people don't study them much except for blood groups and things like that. So I decided to learn more about that before proceeding any further. As it happened, two of the world's experts on these processes right on my nose at WashU, and I just asked to work with them. Looking at the Kornfelds, I studied phosphorylated high mannose oligosaccharides on N-linked glycoproteins. And you can see in those days, it was common to have just two authors on a paper. quite different from today. This work resulted in the elucidation of pathway of lysosomal enzyme targeting lysosomes. Unisynthesized glycoproteins enter the Golgi, and those that get -6 phospholipid get diverted to lysosomes. The others continue along the path. There's a defect in human disease in that pathway. First example, such a difference. I started out my independent career. I decided to look at other such modifications. Sulfate-linked oligosaccharide mammalian cells. With HUD freeze, they found that the glycogen sulfate chains attached to complex type glycans. And as a specific glycoprotein that had these esters on them. This is not really pursued a whole lot more. Not many people have studied this. They need to study sulfation. It's very important in terms of beyond glycosamine glycans. I decided to study sialic acid and cell-secreted molecules. They found the tips of glycolipids, O-glycans, N-glycans, in high density in animals with deuterostomal lineage. And interestingly, the commensal and pathogenic bacteria interact with deuterostome. So the bacteria are coating themselves in a sort of Klingon cloaking device to avoid detection by the human immune system, like coating themselves with sialic acids. The remarkable thing with sialic acids is the amount of diversity in the mammalian glycome. It's the 9-carbon backbone of the so-called non-lysonic acid. And there are many such modifications. Here's a remarkable one I found early on, working with Andel in the days before MALDI mass spec. Here's N-acetylnilaminic acid, the common sialic acid in all systems. Here's one we found, 8-O-methyl 7,9-dioacetyl N-glycosaminic acid. Huge number of changes, and changes specific to cell types and tissues and so on. Another striking feature is the overacetylation 7, 8, and 9 position. The acetyl groups can actually migrate along the chain, the 7 to 9 position under physiological conditions. Also, acetyl groups in the 4 position. And these have various effects on biological systems. Early on, we recognized a monoclonal antibody that recognized overacetyled sialic acid in human melanomas with ganglioside, working with Dave Cherish and others. Found that an acetyl transferase was adding this acetyl group to the GD3 molecule, ceramide, and altering its function and detection in tumors. We then got into studying more of these, the pulse chase studies of the turnover of these acetyl groups, and found that N-acetyl groups were also turning over rapidly. We studied these and found examples of changes occurring in different pulse chase studies. And putting these all together back then, we found that the glycolipid that begins at GM3 can get acetylated in G3, 7 position, 9 position, migration to the acetyl group. And there's likely DNA acetylation yet to be found because we found the product, but not the enzyme yet. This whole area needs a lot more study, which we pursue up to this. And monoclonal antibodies recognize them. Meanwhile, we found that the right liver Golgi apparatus is an excellent system study of acetylation. We studied acetylation, deacetylated sialic acids in various systems and examined them in detail. I'm not going to go into the details here, of course. And we also found esterases that could remove acetyl groups from sialic acids. And a lot more interactions going on in the Golgi apparatus, setting of acetylation. We studied them further, including and then studied acetyl esterase from right liver. This is an ancient summary slide from the 1990s. Still largely accurate, but what we put together here, the transmembrane acetyl transferase, is probably the Cas-T1 protein that has recently been discovered by others. A lot more work to be done on this. We continue to study lysosomal enzyme sialic acid esterase, studied the effect of overacetylation on leukemia complement activation by I-type lectins and tissue homing, and regulation, growth, and differentiation in leukemia cells. Taking advantage of the fact that we are studying intact glycobiology, I also studied glycans labeled by other molecules using radioactive tracer techniques are no longer used. Another modification of sialic acid is the conversion of nu5AC to nu5GC. And we studied this and found that the primary site of hydroxylation in acetylenic acid is cytosolic sugar pool. So here's the figure showing the pathways of turnover of sialic acid in cells. Here you can see that when CMP nu5AC is converted to CMP nu5GC, you have a donor to add GC to molecules. And some of this can be recycled. The remarkable finding that was that CMH was missing in humans, and we could not convert AC to GC. So the first known genetic difference in humans and great apes. Because they have a GC and AC, and we only have AC, an excess of AC. Then we found traces of GC in human tissues. We began to wonder whether diseases are human-specific or related to changes in sialic acid biology. We found several examples. I'm going to go into a few. Here's one example. Plasmodium malaria, plasmodium malaria. It appears that many of the malarias that are circulating in African apes prefer GC for a target. You imagine that what happened at the time of the loss of GC, probably over 2 million years ago, the loss of the nu5GC targets, and humans had a free ride for 1 million years or so from this parasite. The parasite's always been in the end, they came back to bind AC. Now this is exploitation, and we of course are exaggerating things by farming and doing other things. There are many examples like this: typhoid, cholera, mumps, etc. Many diseases that are human-specific appear to be associated with sialic acid changes. All this is genetic changes. But despite the fact that the CMH null mouse had no GC in its body, GC has been found in humans in various tissues, despite anti-GC antibody responses. Basically, we figure out what's going on. GC is not found in poultry, eggs, plants, or fruits, and is low in fish. It's present in mammals in large amounts. When we eat that, we undergo postnatal immunization, induce GC antibodies. And this xeno-autoantigen, as we call it, interacts with xeno-autoantibodies and generates chronic inflammation, xenosialitis, which is part of, we believe, exaggeration of disease caused by consumption of excess red meat. Also, another JBC paper studied the uptake and incorporation of this non-human sialic acid in human cells. Red meat-derived glycan then promotes inflammation, cancer progression, and enhance atherosclerosis by intrinsic extrinsic mechanisms. So it's not just the immune reaction. We found that the loss of GC in humans is associated with many changes, which is typical of humans: hypoglycemia, diabetes, chronic inflammation, LDL changes, reactive macrophages, reactive T and B cells. All of these together, if you have GC-rich food intake, give rise to mutations. Give rise to atherosclerosis aggravation. Just similar mechanisms of accelerating carcinoma progression in humans. More recently, we studied the vertebrate amino sugars, the N-glycyl groups, so very few, though nu5GC is the most common one. Studied turnover and showed that it turns over all the way down to chondroitin sulfate with GC on it. But the only source of GC in the cells is from the CMH. Another class of sialic acid binding proteins is selectins. We studied their ligands in endothelial cells, leukocytes, and platelets. And they interact with selectins in various mechanisms that have been described in the normal state. Invasive carcinomas express mucins that contain ligands for selectins, and this is involved in cancer metastasis, can be blocked by heparin, clinically available heparin. Yet another difference we studied was the linkage in the 2 position, the 6 position. And it turned out that CD22 of B cell-specific Ig supply members with sialic acid binding lectin. It was bound 2 6-linked sialic acids and had a lot of specificity that we worked out and published in JBC. Another set of JBC papers allow us to characterize the sialic oligosaccharides adhesion to cytokine active human endothelial cells, and binding of plasma cytoglycoprotein. So this is so many others in our study, and it's the origins of discovery of a set of proteins that I named the sialic acid binding Ig superfamily lectins, SIGLEX. We have been studying CD22, as I mentioned, that is affecting sialic acid binding at alpha-2,6 linkage. Paul Crocker and I have been studying SIGLEC1, which binds to thrilling sialic acids. These are conserved SIGLEX. But starting out this in genomes, we found many human-related C30-related SIGLEX, all the way down to 16. Actually, the number 17 present in some primates. And these C30-related SIGLEX are related to immune reactions, prominently expressed immune cells. Whereas in mice have many fewer SIGLEX, and it may cause a complication in identifying orthologs as well as the nomenclature. But many animals do have quite a few number of SIGLEX, as we'll see in a minute. Another unusual finding was the change in SIGLEC12 in humans. There's a human mutation of critical arginine residue make it unable to bind sialic acid. So it isn't really a sialic acid binding protein anymore, which is why I had to put a Roman numeral on its number. Chimpanzee SIGLEC12 preferentially recognized GC, the non-human sialic acid. Totally, the humans lost GC by mutation of Nu5-GC of CMH. Restoring the arginine human SIGLEC restored sialic and preferentially to the chimpanzee target, which makes sense. But the SIGLEC gene harbors a common polymorphic frameshift mutation causing truncation of SIGLEX, alternate splicing, loss of expression, full-length protein majority humans. The majority of humans don't actually express SIGLEC12 anymore. Why would you bother to study it then? We think this is what happened, that the human-specific solubilization of CMH, the loss of GC of the binding ligand for great apes, which is Nu5-GC. Humans only produce Nu5-AC, no preferred ligand. And this allowed the addition of this human-specific arginine mutation, which may have also protected from pathogens that are trying to take advantage of the SIGLEC. Multiple polymorphic nonsense mutations. And across populations, eliminate protein expression in some, but not all individuals. Turns out in epithelial cells, SHP2 is an important oncogene. It's not just an individual molecule like it's found in immune cells. About great apes, humans are very prone to advanced carcinomas. Ajit Varki has written a lot about this. In tissue sections, they're both malignant adjacent normal tissue present. This is an individual that has the gene. Actually, the induced even higher in malignant cells. And when this is immunohistochemical analysis of SIGLEC12 on enhanced expression, high frequency of carcinomas. So if you look at normal tissues and samples from a population, you find that normal tissues have SIGLEC12 only in a third of the population, is what you expect from the genetics. But in tumors, most tumors have SIGLEC12, since the gene cannot be repaired. This, we believe, is recruitment of SIGLEC12 into advanced cancers. We found a SIGLEC12 now human carcinoma cell line. The only rare ones that you do find, the accelerated tumor growth in nude mice and enriched transcripted genes of cancer progression. Many of the same genes are involved in other SHP2-dependent cancers. Expression of full-length SIGLEC12 did not correlate with early cancer risk. So high frequencies found advanced colorectal cancer and correlates overall survival, and seems to be a molecule that is driving late stages of cancer progression. So this needs to be studied much further in terms of effects, but in a transgenic mouse system, we found that SIGLEC12 was inducing rapid growth of cancers. Going on to compare them with humans and other apes, we found that most of the SIGLEX are missing in mice. The humans and chimpanzees are very similar, except for one or two that are missing. SIGLEC3 in humans underwent some very unusual changes. It's expressed prominent in human microglia. Seems to control inflammation in the brain. human-specific isoform loss of sialic acid being protected against Alzheimer's disease. Here's a GWAS study. And what we found is that, and others worked on this too, is that the loss of the sialic acid binding domain here results in different forms of CD33, which diverge a different intracellular pool and protects against Alzheimer's disease progression. So how did this come about? In evolution, you can never be sure of anything except what you can prove indirectly or show that the facts fit. But it appears that, well, Neisseria species are common non-pathogenic airway common source. Only in humans, they evolved into human-specific deadly pathogens, gonorrhea and meningococcal meningitis. And it's an acquired state that a few bacteria have taken advantage of the fact that humans do not express GC, only express AC. Another interesting thing relates to post-reproductive lifespan. If you look at long-lived animals like Asian elephants or chimpanzees, as their fertility goes down, they start dying. And according to evolutionary theory, you should not be around anymore after you stop reproducing. But both in hunter-gatherer tribes and in the killer whale, we found that the lifespan is much longer after the loss of fertility. So these infertile grandmothers who have gone through menopause can care for their grandchildren. So the idea is that the grandmother hypothesis tells you that humans are able to take care of larger numbers of small, helpless babies because of infertile elders, grandchildren, and grandmothers. Here's an example of evolution scenarios that we concoct. Some of it is speculative, some of it is true. We try to put it together eventually to get the right answer. I'm going to go into this complicated slide, which indicates many steps that occurred in the evolution of CD33. So here's a figure showing what happened. Yeah. Chimpanzees have a functional CMH and CD33 binds mu5GC as the self. Mammalian only have mu5AC. Loss of CMH in humans allow CD33 now bind AC. And it now gets expressed in microglia and binds mu5AC in the brain. In some people, exploitation by pathogens occurs, and then we see there is a truncated variation, evading pathogen manipulation. We have more active microglia. This truncated form of CD33 seems to protect against Alzheimer's. We're actually finding several genes that protect against late life cognitive disorders. And this seems to be because we want to prolong the life and functionality of grandmothers. Another thing had to do with lifespan. Found that the cyclic receptors can modify lifespan by modulating oxidative stress. And here we're looking at the number of cyclic genes. Cyclic genes tend to duplicate, and different species have different numbers of cyclic genes. You look at them and you see this amazing correlation between the number of cyclics and the lifespan, maximum lifespan. In other words, if you have more cyclics, you live longer, or if you live longer, you evolve more cyclics, which is the case. But humans fall off the line here. We more recently looked at many other species and found that this correlation is really strong, except for orcas and humans. But if you put them on the reproductive lifespan, they fit exactly on this pathway. And this suggests that oxidative events are involved in aging. Uniquely human genetic changes occur in cyclic genes. I won't go through them in any great detail. Mentioned a couple of them. Most of these are not found in apes, including some deletions and some other changes that have occurred in human evolution. Also, uniquely human change expression of cyclic genes. CD33 I mentioned is Alzheimer's disease, cyclic 5, gene controlling lymphocyte activation, HIV, progression to AIDS, which occurs in humans, but not so much in chimpanzees. Cyclic 5 and 14 are restored the sialic acid binding property on amniotic epithelium and visited intrauterine infection of other human-specific pathogens, group B strep. Also found in the placenta, cyclic 6 found only in the human placenta trophoblast, not in the other apes. And it's increased further in human disease called preeclampsia. Cyclic 7 is associated with pancreatic islet cells only in humans, not chimpanzees. Maybe associated with type 1 diabetes. Cyclavin 16 expressed in the brain, only in humans, and may facilitate E. coli K1 meningitis, because E. coli K1 makes a similar ligand. Also, expression in the ovarian fibroblasts is different in humans and chimpanzees. So I've told you about sialic biology as a hotspot in human evolution and disease, and many changes that have occurred. And this is obviously going to keep us busy for a long time. In fact, we're like kids in a candy shop. There's a lot more to be studied. We hope others will join us. Circling back to this, where I started, about the importance of research in medicine and the importance of physician scientists participating in this research. Another famous physician scientist, Arthur Kornberg, wrote this article once. He wrote several articles about this, pointing out that key contributions to diagnosis, treatment, and prevention of cardiovascular disease, pulmonary disease, this particular case that have been studied. 2/3 have been due to basic research finding rather than applied research. So I think we need to continue the philosophy that is now being fading away of having physicians, a small subset of physicians, go into basic research and they can understand the disease and cross-fertilize between the systems. With that, I'll end and thank you for your time.